Nice. High tech. Okay. Hello. All right. Pipette. My raison d'etre. So here I am pipetting stuff. You know why I'm pipetting right now?
Why are you pipetting?
Because I didn't do a very good job pipetting last time.
Oh no.
So I have to fix it. F2 goes in F2. It's kind of like Battleship, only with DNA.
So what is it you're doing right now?
What I'm doing right now is you may or may not have been able to see inside these wells there's this sort of muddy little spots, and the muddy little spots are little microscopic magnetic marbles.
Hold it up again for a second.
Yeah.
Let me zoom in.
And if you remember the marbles that had like swirly stuff in them, so the coloring is the swirly stuff, and on the outside it is silica, which is glass, right? So it's glass just like a real marble with real stuff in it, only the stuff is magnetic. And it can be attracted to a magnet. The silica on the outside binds DNA pretty well. So we were using it, you mix it with your cell lysate, and then all the DNA sticks to the little marbles, right? And then you bring the marbles down with these incredibly powerful magnets, which would probably, I'd bring them close to your electronic equipment to show you, but then it would probably ruin the electronic equipment. So anyway, right, yeah, so so after you've got your cell lysate, which is a bunch of cell guts and DNA, and all we care about is the DNA. So I used this buffer and a bunch of buffers, and I washed my DNA, right? The DNA was stuck to my metal marbles, and then once I was done, I used this last one right here in this elution buffer, right? And this stuff, when you pour it in, it actually, the DNA likes the elution buffer better than it likes the marbles. So now it lets go of the marbles, floats in the solution, and I slurp it up with my raison d'etre here. Is that clear? Good. I'm a teacher, you know, so if it wasn't clear, I'd get fired.
And so what are these little DNAs? Why are you trying to do that?
Oh, yeah. So what is the DNA in each of these things? I could explain in general terms, but not in specific terms. In general terms, they are little DNA parts, because this is synthetic biology, where DNA has been converted. Part of the purpose of synthetic biology is to make biology easier to do, and so what we've done is tricked out each little natural DNA sequence with sequences at the beginning and the end of those sequences of DNA that makes those sequences easy to cut. And when they're easy to cut, they're easy also to glue back together, which basically effectively turns each of these little pieces of DNA into a Lego piece that we can stack into a bacterium that will make the bacterium do stuff, like respond to sound and mysteriously commit suicide after hearing Beethoven's Ninth Symphony, and other things that will impress and amuse judges and may ultimately prove useful to society at large.
It should be lined up, right?
Right. So in terms of specifics, though, I can't really tell you. This is what you call a 96 well plate. And if you've ever heard the two words high throughput put together, what it means is that what you are looking at here is sort of an industrialized process. And for the past week, I have been the mini prep guy. And a mini prep is basically
Like the fingers will be perfectly aligned.
pipetting out only a certain fragment of DNA, a certain, not just like the parts, but something called a plasmid, which is sort of like a Lego kit, right? Legos are interchangeable from one kit to the next kit, but this is like the castle kit in this one, and this is like the, you know, fire engine kit, and this one is like the, you know, the other Lego kits that you may or may not have had your parents purchase when you were a kid. Anyway, so because this is high throughput, I actually have no idea what I'm pipetting. I know there's DNA in there, but you know, actually, in fact, it may be worthwhile to point this out. This magnetic bead stuff is not intended for use by humans. It's actually intended for use by a robot. So as Aaron points out, I actually am a robot.
That was great.
So when the camera's off, I'll be plugged back into the wall for maintenance. But I'm actually a robot that screws up a rather high frequency of the time, but not today. No siree. Look at that. Look at that. Perfect. Did you have any more questions?
This is so entertaining.
I've been waiting a long time to talk about this.
I have a question.
Yes, in the back. Aaron, I'm sorry. One more time, please.
Why aren't you wearing pants?
Aaron, you asked, why am I not wearing any pants? I'm actually wearing a really nice lab coat. And I actually...
Can you do a walk?
Oh,
over there?
you want the walk? Remember, I am mic'd, so I don't want to drag you and your camera about.
Okay, right there.
Always the beats off by it. Yeah,
Why am I like this? I don't know.
It's the magnets that did it too.
They say EMFs are rather harmful to one's mental state. You ever see that? Have you seen that that YouTube with the cell phones?
No.
Oh, you haven't seen this? So you get 4 cell phones, put them together, aim the antennas all towards the center point, take several unpopped grains of popcorn, place them in the center, call the phones, and guess what happens?
The popcorn pops?
That is correct.
Wow.
Yeah, so all that hooey about, cell phones cause cancer, and no, they don't cause cancer.
They just make popcorn.
They just make popcorn, and presumably when you hold them close to your head and the electromagnetic field is making, popcorn out of your
I hope mine's kettle
brain.
corn. I prefer that.
It'll be more delicious for those for your loved ones. What's that?
Sell it at the street fair.
Oh yeah, so I had a friend who bought a mouse to feed